Hello, everyone. We are the last group, and we will be reporting about the second to the last exercise that we will have this semester, which is exercise 13, the metabolism of carbohydrates and organic acid. The authors of the report are Kimberly Bantolio, Shane Mariel Caba, Sheena May Cabugnason, Sophie Rhea Maglangit, Will Geraldine Vicio, and yours truly, Christina Cassandra Pava.
Good day, everyone. So, for today, we will be conducting an experiment on exercise 13 entitled Metabolism of Carbohydrates and Organic Acids. So, please bear with us, and without further ado, let's start. What is metabolism? So, metabolism refers to all the biochemical reactions that occur in a cell or organism. The study of bacterial metabolism focuses on the chemical diversity of substrate oxidations and assimilation reactions, or reactions by which substrate molecules are broken down, which normally function in bacteria to generate energy. Also, within the scope of bacterial metabolism is the study of the uptake and utilization of the inorganic or organic compounds required for growth and maintenance of a cellular steady state or assimilation reactions. These respective exergonic or energy yielding and endergonic or energy requiring reactions are catalyzed within the living bacterial cell by integrated enzyme systems, the end result being self replication of the cell. The capability of microbial cells to live, function, and replicate in an appropriate chemical and the chemical changes that result during this transformation constitute the scope of bacterial metabolism. Bacteria metabolize carbohydrates via either respiration, aerobic or anaerobic, or fermentation. So these are the heterotrophic bacteria, which include all pathogens, obtain energy from oxidation of organic compounds. Carbohydrates, particularly glucose, lipids, and protein, are the most commonly oxidized compounds. Biologic oxidation of these organic compounds by bacteria results in synthesis of ATP as the chemical energy source. This process also permits generation of simpler organic compounds needed by the bacterial cell for biosynthetic or assimilatory reactions. All heterotrophic bacteria require preformed organic compounds. They are the most commonly studied bacteria because they grow readily in media containing carbohydrates, proteins, or other complex nutrients such as blood. Cellular respiration is a process that takes place inside the cells where energy is released by the breakdown of glucose molecules. The process can be conveniently divided into two categories based on the usage of oxygen, namely aerobic and anaerobic respiration. As already stated, cellular respiration can be of two types, aerobic and anaerobic. Aerobic means with air. Therefore, aerobic respiration is the process of cellular respiration that uses oxygen to produce energy from food. Anaerobic means without air. Therefore, this type of cellular respiration does not use oxygen to produce energy. Sometimes, there is not enough oxygen around for some organisms to respire, but they still need the energy to survive. Due to lack of oxygen, they carry out respiration in the absence of oxygen to produce the energy they require, which is referred to as anaerobic respiration. The fundamental difference between aerobic and anaerobic respiration is the usage of oxygen in the process of cellular respiration. Aerobic respiration, as the name suggests, is the process of producing the energy required by cells using oxygen. The byproduct of this process produces carbon dioxide along with ATP, or the energy currency of the cells. Anaerobic respiration is similar to aerobic respiration, except The process happens without the presence of oxygen. Consequently, the byproducts of this process are lactic acid and ATP. There are different tests used to metabolize carbohydrates and organic acids. First, we have Hugh and Lipson test, which is used to determine whether an organism uses carbohydrate substrates to produce acid byproducts. The medium contains rich source of carbon and also contains pH indicator dye, bromothymol blue, which is green or blue under alkaline oxidative conditions, or yellow under acidic fermentative conditions. Non-fermentative bacteria are routinely tested for their ability to produce acid from six carbohydrates, such as glucose, xylose, mannitol, lactose, sucrose, and maltose. Whether an organism is oxidative or fermentative can be determined by using Hugh and Lipson's medium, or commonly called as OF medium, which contain tryptone and bromothymol blue as an indicator. Two tubes are required for interpretation of the OF test. Both are inoculated and one tube is overlaid with Vaspar, while the other is not. The Vaspar serve as a barrier to oxygen to help achieve an anaerobic environment. Growth of microorganisms in this medium is either by utilizing the tryptone, which results in an alkaline reaction or dark blue color, or by utilizing glucose, which results in the production of acid or turning bromothymol blue to yellow. Production of acid in the overlay tube and open tube results in a color change and is an indication of fermentation. Acid production in the open tube and color change is the result of oxidation. Some bacteria have the ability to utilize glucose and convert it to stable acid like lactic acid, acetic acid, or formic acid as the end product. These bacteria initially metabolize glucose to pyruvic acid, which is further metabolized through the mixed acid pathway to produce the stable acid. The type of acid produced differs from species to species and depends on the specific enzymatic pathways present in the bacteria. The acid so produced decreases the pH to 4.5 or below, which is indicated by a change in the color of methyl red from yellow to red. In the methyl red test or MR test, the test bacteria is grown in a broth medium containing glucose. If the bacteria has the ability to utilize glucose with production of a stable acid, the color of the methyl red changes from yellow to red when added into the broth culture. In the Fogges-Proskauer or VP test, is used to determine if an organism produces acetylmethylcarbinol from glucose fermentation. If present, acetylmethylcarbinol is converted to diacetyl in the presence of alpha-naphthol, strong alkali, and atmospheric oxygen. The alpha-naphthol was not part of the original procedure, but was found to act as a color intensifier by Barrett and must be added first. The diacetyl and quinidine containing compounds found in the peptones of the broth then condense to form a pinkish red polymer. The methyl red and Fogges-Proskauer test are frequently employed for identification of enteric bacteria that include Klebsiella, Enterobacter, and Escherichia coli. Citrate agar is used to test an organism's ability to utilize citrate as a source of energy. The medium contains citrate as the sole carbon source and inorganic ammonium salts as the sole source of nitrogen. Bacteria that can grow in this medium produce an enzyme, citrate permease, capable of converting citrate to pyruvate. Pyruvate can then enter the organism's metabolic cycle for the production of energy. Growth is indicative of utilization of citrate, an intermediate metabolite in the Krebs cycle. When the bacteria metabolize citrate, the ammonium salts are broken down to ammonia, which increases alkalinity. The shift in pH turns the bromothymol blue indicator in the medium from green to blue above pH 7.6. An organism that simultaneously can utilize sodium malonate as its carbon source and ammonium sulfate as its nitrogen source produces an alkalinity due to the formation of sodium hydroxide. The medium used for malonate utilization test is malonate broth. It contains mineral salts, sodium malonate for carbon, and ammonium sulfate for its nitrogen source. The pH indicator is bromothymol blue, which is green at neutral pH, yellow at acidic pH, and turns blue at alkaline pH. Organisms which simultaneously utilize malonate and ammonium sulfate produce sodium hydroxide, which thereby results in an alkaline reaction and changes the indicator from its original green color to light blue or Prussian blue. Organisms which cannot utilize malonate and ammonium sulfate and do not ferment dextrose produce no color change. Organisms which are malonate negative but do ferment dextrose result in the development of a yellow color due to increased acidity in the medium. Many bacteria produce extracellular enzymes used to catalyze chemical reactions outside of the cell. In this manner, nutrient sources such as starch, that are too large to be absorbed through the cell membrane, can be broken down into smaller molecules and transported into the cell via diffusion. In the starch hydrolysis test, the test bacteria are grown on agar plates containing starch. If the bacteria have the ability to hydrolyze starch, it does so in the medium, particularly in the area surrounding their growth, while the rest of the area of the plate still contain non-hydrolyzed starch. Since no color change occurs in the medium when organisms hydrolyze starch, iodine solution is added as an indicator to the plate after incubation. While the non-hydrolyzed starch forms dark blue color with iodine, its hydrolyzed end products do not acquire such dark blue color with iodine. Consequently, transparent clear zones are formed around the colonies that hydrolyze starch, while the rest of the plate show a dark blue coloration as iodine forms the colored complex with starch. Gluconate test medium is used in detection of gluconate oxidizing microorganisms. This medium is used to check the ability of an organism to oxidize gluconates, the sole carbon source, to the reducing compound 2-ketogluconate, which subsequently accumulates in the medium. Peptone and yeast extract provides nitrogen and other nutrients necessary to support bacterial growth. The potassium hydrogen phosphate buffers the medium. The basis of the test is the change from gluconate, a non-reducing compound, to 2-ketogluconate, a reducing compound, which is tested using suitable reagent. The cupric ions in the reagent are reduced to cuprous ions by free ketone group of 2-ketogluconate, and an orange-yellow precipitate of copper oxide results. At the end of this exercise, we should be able to identify the type of carbohydrate metabolism, such as oxidative or fermentative, of an unknown bacterium using Hugh and Lifson medium. Also, to determine the glucose fermentation pathway utilized by a bacterial isolate through metal red and Fogges-Proskauer test. To determine the ability of a bacterial isolate to utilize citric acid and malonate. To determine the ability of a bacterial isolate to hydrolyze starch, and lastly, to determine the ability of a bacterial isolate to produce 2-ketogluconate from potassium gluconate. These are the materials used for this exercise. We have the bacterial cultures such as Bacillus subtilis, Enterobacter aerogenes, Escherichia coli, Pseudomonas fluorescens, and unknown bacterium. The culture media used for this exercise are Haynes medium, Hugh and Lifson with different sugars such as glucose, lactose, mannitol, sucrose that are sterilized separately, malonate broth, nutrient broth plus 0.5% glucose tube, Simon citrate agar slant, and starch agar plate. In addition, we use an empty sterile test tubes, glass pipettes, sterile Petri plates, and test tubes. These are the reagents used on this exercise, we have Benedict's reagent, Lugol's or Graham's iodine, methyl red indicator, and freshly prepared Fagus Prescower or VP reagents. So these are other materials used on this exercise. We have disinfectant or quaternary ammonium compounds, medicine droppers, pipette aid or rubber bulb, pipetor, sterile white tips with 5 ml capacity, sterile water agar 2% Vespar, test tube rock, water bath, wire loop, and wire needle.
Good day, everyone. I am Bantolio Kimberly Dita. For the continuation of our report, I'm going to tackle about the oxidation fermentation. Oxidative fermentation test. It is used to determine if bacteria metabolize carbohydrates oxidatively by fermentation or are non-saccharolytic and therefore have no ability to use the carbohydrate in the media when you culture a bacteria. So for the reagents and equipment that are going to use in the experiment, I prepared discrete bacteria colonies growing on a solid medium or prepared. Then two different media used in depending on the organism that being tested. So the bacteria that are going to be used in this experiment is Staphylococci and Micrococci. So these two um, family species of bacteria require Baer Parker's modification of the medium. And then for the gram-negative rods, it will require Hugh and Lifson of basal medium. So using this medium, Hugh and Lifson, we were able to differentiate gram-negative bacteria into three categories, which is for fermentative, oxidative, and inactive. So we're going to use also the bacteriological straight wire loop or disposable alternative, then soft paraffin oil or some other use Vaseline oil as substitute for soft paraffin oil. So these are the quality control organisms that use for the gram-negative rods. So for the positive control in oxidation, Pseudomonas aeruginosa, the quality control organism that is going to use is NCTC 10662 or NCTC 12903. So for the fermentation is Escherichia coli. The control organism that is going to use is the NCTC 10418 or NCTC 12241. And then for the negative control, so there is no reaction. So the species of bacteria that is going to use is the Acinetobacter ewolfii. The control organisms that is going to use is NCTC 5866. So for the gram positive cocci, for the positive control oxidation, the species of bacteria that is going to use is Micrococcus luteus, and the control organism is the that are that is going to use is NCTC 2665. And then for the fermentation, the species of bacteria that is going to use is Staphylococcus aureus. And the control organism that is going to use also is NCTC 6571 or NCTC 12973. And then for the negative control, there is no reaction uh, of basal medium without carbohydrate. So no control organisms that is going to use in this. For the first step, we're gonna isolate discrete colony. So we're gonna pick at the top of the single colony of organism, and then streak it into into the given media to culture bacteria. So after that, we're gonna heat 2 tubes of medium in boiling water for 10 minutes to remove the oxygen and cool it down. So it was shown in the picture how you are going to um, boil to heat tubes in a boiling water. Then after that, the organism to be studied is inoculated in two long narrow tubes, 12 by 120 mm by deep stab inoculation. So in inoculation, we're gonna stab it and then with the use of straight wire. So after that, one tube is covered with oil or a Vaseline layer or the soft paraffin oil to induce an aerobic to environment that forces the strain to carry out fermentation. Then after that, we're gonna incubate it at 35 degrees Celsius for 48 hour or longer. Some uses 24 hour, 24 hour, but in this case, we're gonna use 48 hour or 48 hour. So in the tube one, it was marked aerobically, and the tube two will be anaerobically, which is the where we use the soft paraffin oil or the Vaseline for the layer to induce an aerobic environment. So for the interpretation, so in gram-negative rod reactions, positive resolve and oxidation, acid in aerobic tube only yellow color, and aerobic tube then green in anaerobic tube. Then for the fermentation, the acid in both tubes is called yellow color. And then for the negative result, neither fermentation nor oxidation, no acid production or blue or green color in aerobic tube, green in anaerobic tube. So for the gram-positive Coca reactions, the positive result of oxidation is acid in aerobic tube only yellow color in aerobic tube, purple in anaerobic tube. While in the fermentation, acid in both tubes, same and gram negative, it has only yellow color. So for the negative result, neither fermentation nor oxidation, no acid production or no color change, purple color in both tubes. So note the semi-solid consistency of the medium also allows for detection of motility. Hazy growth, away from the stab line can also be noted. So this slide shows the uh, overall product or the overall result of the experiment. So oxidation, acid in tube 1 only. So in tube 1 yellow color, in tube 2 green color. So gram negative rod, this is the gram negative rod reaction. And then for the tube 1 yellow color and tube 2 purple color, so this states the gram positive cocci reaction. And then for the fermentation, acid in both tubes yellow color, and then the gram negative rod and the gram positive cocci is react. So neither fermentation nor oxidation, so no acid production. So in the tube 1 blue or green color, so in the tube 2 green color. So in gram negative rod, this is the gram negative rod reaction, while the both tubes in the has a purple color. This is the gram positive cocci reaction. So this is the end of my report.
Good day everyone, I'm Sheena Kabugnason and now I am going to discuss the methyl red test and the Voges-Proskauer test. So when glucose is fermented, a variety of products are possible. This is a test used to determine some of those products. Most organisms differentiated by this test convert the glucose to acids. Some bacteria continue to produce more acid in mixed acid fermentation. Some genera use the glucose but produce end products with a more neutral pH like those that result from butanediol fermentation. And the purpose of this test is to distinguish between members of the family Enterobacteriaceae, but now they are used to characterize other groups of bacteria, including actinobacteria. In this test, we are going to use dropper, methyl red indicator, 5% alpha-naphthol, 40% potassium hydroxide, and bacterial cultures of Escherichia coli and Enterobacter aerogenes. So its media also contain 7 grams of polypeptone, 5 grams of glucose, 5 grams of dipotassium phosphate, 1 liter of distilled water, and its final pH is 6.9. So let us first understand the theory behind methyl red test. Methyl red is a pH indicator that changes colors when fluctuated between different pH. In a methyl red test, what we are testing is the ability of bacteria to convert pyruvic acid after glycolysis to mixed organic acid. So in this diagram, starting from glucose, it will go through glycolysis to form pyruvic acid. Then, the pyruvic acid will convert to the mixed organic acid. So as we start with glucose, the methyl red is yellow in color. But as we go through glycolysis to form pyruvic acid and then further mixed to organic acid, the mixed organic acid are going to titrate methyl red with the decreasing pH to a red color. So if we're doing a methyl red, then we see a red color develop. That means that the bacteria was actually able to convert pyruvic acid into mixed organic acid after glycolysis. And one thing we should note is that the result of the methyl red are very similar to the result of the Voges-Proskauer test. So if it is yellow in color, meaning these bacteria do not have the ability to convert pyruvic acid to mixed organic acid. We know that glucose can be converted into pyruvic acid through the process of glycolysis. When we look at the methyl red test, the pyruvic acid could be converted to the next organic acid, And if it turns red, it is going to be a positive methyl red test. But in Vogg's Prescower test, it is something a little bit different. It turns out that pyruvic acid can be converted ultimately through a series of two enzymes to a molecule called acetone. Now, acetone is not directly what we're measuring, but it turns out that acetone is involved in the series of metabolic transformations. We can form various compounds like diols or diones. So, if we have a bacteria that can produce acetone, what we can actually do is put him a series of reagents, and we can convert the acetone in the absence of enzymes to diones called diacetyl. And diacetyl can react with peptones, specifically arginine residues of peptones, to produce a red color. So if we put alpha naphthylamine and potassium hydroxide with oxygen gas, acetone can be converted without the need for an enzyme into this dione called diacetyl. In this mixture, we also have peptone, and it contains arginine residues. It turns out that arginine can react with diacetyl, and when it does, the color will be changed into red. So what the Vogg's Prescower test is measuring is the ability of the bacteria to convert glucose into neutral end products. Diacetyl and butanediol are both neutral end products. However, we're specifically reacting these reagents with the acetone because the acetone is indicative of its ability to convert glucose to neutral end product, and a red color change could be a positive to Vogg's Prescower test. And if it's red, that means that the bacteria are able to convert glucose into these neutral end products such as acetone and diols and diomes. And if we don't see the reddish or purple color change, that is a negative result, and so the bacteria do not have this ability. So for the procedure, inoculate E. coli and E. aerogenes to two test tubes of MR VP broth using a wire loop. Then, incubate at least 33 Celsius for 48 to 60 hours. So for the methyl red test, transfer 1 ml of the culture to a tube and add 1 to 2 drops of methyl red indicator. So red color is positive for MR test, indicating acid production from glucose, and then yellow is negative. So for Vogg's Prescower test, to the rest of medium, add 5 drops of 5% of alpha naphthol and mix. Then add 7 to 10 drops of 40% potassium hydroxide. Then mix and stand for 15 to 20 minutes. So a pink to red color is positive for Vogg's Prescower test reaction, indicating acetone production. So if negative, there will be no change at all. So as a result, Escherichia coli is positive in methyl red but negative in Vogg's Prescower test. So it means that E. coli has the ability to convert pyruvate to mixed organic acid, and but it does not have the ability to convert glucose into neutral end products. And E. coli is also called as mixed acid fermenter because it produced a mixture of fermentation acids and thus acidify the medium. While Enterobacter aerogenes is negative in methyl red but positive in Vogg's Prescower test. It only means that E. aerogenes has the ability to convert glucose into neutral end products, and it is also called as butanediol fermenter because it forms butanediol, acetone, and fewer organic acid. So the pH of the medium does not fall as low as during mixed acid fermentation. At a pH of 4, the methyl red indicator turns red, and it is a positive methyl red test. And at a pH of 6, the indicator turns yellow. so a negative methyl red test. Also, E-orogenase doesn't have the ability to convert pyruvate into mixed organic acid.
Good day, everyone. I am Christina Cassandra Pava, and I will be reporting about the citric acid utilization. So, the procedure for this test includes inoculating an E. coli, which is a gram-negative bacterium, Enterobacter aerogenes, which is a gram-positive bacterium, and the unknown bacterium to the three Simon citrate agar slants using a wire loop by stabbing the butt and streaking on the surface. Next, the Simon citrate agar will be incubated at 33 degrees Celsius for 48 hours. So the expected results will be if it is a positive test, it will demonstrate a growth with a color change from green to intense blue along the slant. If it has a negative result, it demonstrates by no growth and no color change, and the color of the slant remains green. So for the interpretation of the results, E. coli yields a negative reaction, which means the color of the medium remains green. This means that E. coli do not use sodium citrate as a source of carbon and inorganic ammonium hydrogen phosphate as its source of nitrogen. Like many organisms, E. coli has a citric acid cycle, and so it metabolizes citrate while growing on various substances. However, it cannot grow aerobically on citrate because it does not produce a transporter protein when oxygen is present that can bring the citrate into the cell. So on the other hand, Enterobacter aerogenes in the unknown bacterium yields a positive reaction, in which we can see that there is a change in the color of the medium from green to blue. It is capable of fermenting citrate, and it utilizes sodium citrate as a source of carbon and inorganic ammonium hydrogen phosphate as its source of nitrogen. When an organic acid such as citrate, like in the Krebs cycle, is used as carbon and energy source, alkaline carbonates and bicarbonates are produced ultimately. In addition, ammonium hydroxide is produced when the ammonium salts in the medium are used as a sole nitrogen source. So the utilization of exogenous citrate requires the presence of citrate transport proteins or permeases. Upon uptake by the cell, citrate is cleaved by citrate lyase to oxaloacetate and acetate. The oxaloacetate is then metabolized to pyruvate and carbon dioxide. So further metabolic breakdown is dependent upon the pH of the medium. First, under alkaline conditions, pyruvate is metabolized to acetate and formate, producing pyruvate equals acetate plus formate. At pH 7 and below, or the acidic conditions, lactate and acetone are also produced, where pyruvate is equal to acetate plus lactate plus carbon dioxide, and pyruvate is equal to acetone plus carbon dioxide. The carbon dioxide that is released will subsequently react with water and the sodium ion in the medium to produce sodium carbonate, an alkaline compound that will raise the pH. In addition, ammonium hydroxide is produced when the ammonium salts in the medium are used as the sole nitrogen source. So growth usually results in the bromothiamyl blue indicator turning from green to blue. The bromothiamyl blue pH indicator is a deep forest green at neutral pH, with an increase in medium pH to above 7.6. Bromothiamyl blue changes to blue. So the expected results in citrate utilization test, citrate positive if growth will be visible on the slant surface and the medium will be an intense Prussian blue. The alkaline carbonates and bicarbonates produced as byproducts of citrate catabolism raise the pH of the medium to above 7.6, causing the bromothiamyl blue to change from the original green to color to blue color. Citrate negative is when the trace or no growth will be visible. So no color change will occur and the medium will remain the deep forest green color of the uninoculated agar or the E. coli in our test. So only bacteria that can utilize citrate as the sole carbon and energy source will be able to grow on the Simon's citrate medium. Thus, a citrate negative test culture will be virtually indistinguishable from an uninoculated slant. Here in this slide is shown the list of bacteria which gives positive citrate utilization test. Some bacteria also show a variability in the citrate test, wherein different strains give different results. And the other is the list of negative reactions of citrate tests from this given bacteria. So next, we will be discussing the malonate utilization test. So the procedure for this test includes inoculation of the Pseudomonas fluorescens, which is a gram-positive bacteria, E. coli, which is a gram-negative bacteria, and the unknown bacterium to 3 tubes of malonate broth. Then it will be incubated at 33 degrees Celsius for 48 hours. So the expected results will be a positive malonate test which indicates the development of a blue color in the medium, and a negative malonate test is indicated by the media remaining green or turning yellow due to dextrose fermentation. So for the interpretation of results for the malonate utilization test, we can see in this slide that E. coli yields a negative reaction in which the color of the medium remains green. So it can be concluded that E. coli does not utilize malonate as its sole source of nitrogen and carbon. On the other hand, Pseudomonas fluorescens and the unknown bacterium yields a positive reaction in which there is a change in the color of the medium from green to blue. So this means that this bacterium utilizes malonate as sole source of nitrogen and carbon. Malonate utilization test is a colorimetric or differential test of the ability of bacteria to use malonate as source of carbon, the endpoint of which is the production of the alkaline metabolites that induce a color change, which is from green to blue. An organism that simultaneously can utilize sodium malonate as its carbon source and ammonium sulfate as its nitrogen source produces an alkalinity due to the formation of sodium hydroxide. This results in an alkaline reaction which in a medium containing malonate changes the indicator, which is bromothiamyl blue, from its original green color to light blue or Prussian blue. Organisms which cannot utilize malonate and ammonium sulfate do not ferment dextrose produces no color change. Organisms which are malonate negative but do ferment dextrose results in the development of a yellow color due to increased acidity in the medium.
Once again, good morning everyone. Now let's go over to starch hydrolysis test. Many bacteria produce extracellular enzymes used to catalyze chemical reactions outside of the cell. In this manner, nutrient sources such as starch that are too large to be absorbed through the cell membrane can be broken down into smaller molecules and transported into the cell via diffusion. The media that we'll be using for starch hydrolysis test is starch agar. It is a simple nutritive medium with starch added. Beef extract and pancreatic digest of gelatin provide nitrogen, vitamins, carbon, and amino acids. Agar is the solidifying agent and starch is the carbohydrate. Now for its composition, the starch agar is composed of peptic digest of animal tissue, sodium chloride, yeast extract, beef extract, soluble starch, agar, and its final pH at 25 degrees Celsius, 7.4. Going over to the procedure, first let's divide the starch agar plate into 3 sections by marking the bottom of the plate. Second, Let's trick Bacillus subtilis, E. coli, and the unknown bacterium to the 3 section. Next is incubate them at 33 degrees Celsius for 24 to 48 hours. And lastly, flood them with iodine and observe. The positive result will give a clear area around the line of inoculation, and if it is negative, it is blue to black. For our expected result and interpretation, the results of the Bacillus subtilis and the unknown bacterium are both positive. They produce a clear zone around the growth, and they both produce the enzyme amylase which hydrolyzes starch in the agar. Meanwhile, the Escherichia coli is negative on starch, and this means that amylase is absent.
Hello everyone. Speaking right now is Sophie Rhea A. Maglangit, and I will be talking to you about the 2-ketogluconate production. This test is used in detection of gluconate oxidizing microorganisms. In this test, we use Haynes medium to check the ability of an organism to oxidize gluconates, the sole carbon source, to the reducing compound 2-ketogluconate, which subsequently accumulates in the medium. Now let's go over in how to conduct this experiment. First, we inoculate each of the organism, the E. coli, the P. fluorescens, and the unknown bacterium into the Haynes medium tube. After that, we incubate it at 33 degrees Celsius for 48 hours. Then, we add 1 ml of Benedict's reagent to 1 ml of the liquid culture in a tube. These tubes will be boiled in boiling water for 10 minutes and will be cooled rapidly. The development of brown, orange, or yellow precipitate means potassium gluconate was oxidized to potassium 2-ketogluconate. The positive result of this test will produce yellow, brown, or orange precipitate, while the negative result will remain its blue color due to the reagent, or it might appear green. So as you can see in the screen, these are the following results. E. coli test negative, no color change, and the medium remains blue or bluish green. The Pseudomonas fluorescens test positive with a yellow to orange red precipitate, and our unknown bacterium test positive having a yellow to orange red precipitate. So how does this happen? Peptone and yeast extract that are in the medium provides nitrogen and other nutrients necessary to support the bacterial growth. Dipotassium hydrogen phosphate buffers the medium. The basis of the test is the change from gluconate, which is a non-reducing compound, to 2-ketogluconate, a reducing compound, which is tested using a suitable reagent, which is the Benedict's reagent.